Okay, it is Friday. Yes, holiday. It's a 3-day weekend. I won't see you again for 5 days. Oh, yeah, there we go. Yeah. I feel the same way. No, that's not true. I love you guys. I really do. I'm joking. Let's see. We are a little bit behind, and so we have really two options. We can either go faster, or we can do a song. Okay, that's what I thought. So, where are we? Let's see. All right. So, the song we're going to sing is, I try to make the songs relevant, and this song is about a very, very important electron carrier. And it's to the tune of an old Beatles song called Penny Lane. It's not real easy to sing, but that's never stopped me, and I hope it will not stop you. Okay? It's called NAD. In the catabolic pathways that our cells employ, oxidations help create the ATP, while they lower Gibbs free energy, thanks to enthalpy. If a substrate is converted from an alcohol to an aldehyde or ketone, it is clear those electrons do not disappear. They just rearrange. Very strange. NAD is in my ears and in my eyes, helping molecules get oxidized. Voila, NADH, yeah. And the latter is a problem anaerobically, 'cause accumulations of it muscles hate. They respond by using pyruvate to produce lactate, which the citric acid cycle, which the Cori cycle handles transhepatically. I can't remember my cycles. Synthesizing sugars almost effortless, using gluconeogenesis, anabolically, eagerly. NAD is in my ears and in my eyes, helping molecules get oxidized. Voila, NADH. That's it. Okay. A simple one. Now, no. Now, maybe we're behind, but hopefully we feel a little bit better, right? So, okay. So, being behind is not a problem. I will speed up or something. I don't know. All right. So, we're getting ready to talk about electron transport. And of course, I've set the stage for it by talking about the mitochondria, the mitochondrial structure. And so what I want to do here is dive into the actual process. You probably have seen this in other classes. We're going to talk about it at a little bit deeper level, probably than you've seen in some classes. And it's important to keep this in mind. Now, I like to show this figure to start everything off because it makes the problem look a lot worse than it really is. All right? So let's think about what has to happen. So along glycolysis and the citric acid cycle, we've been producing reduced electron carriers. Those reduced electron carriers include NADH and FADH2. And I've been talking on and on, almost incessantly, about the fact that we've got to recycle these guys back to NAD and FAD. So what the electron transport system is going to allow us to do is to recycle those back. So what's going to happen in the electron transport system is that NADH and FADH2 are going to drop off their electrons and protons and become NAD and FAD. Now, in order for this process to happen, it is essential that electron transport be working. That is, that the protons won't get dropped off if the electron transport system is plugged up. And we'll see, not today, but we'll see later how the system can get plugged up. So I want to emphasize that. That's part of something that's very important, control and metabolism. Because if electron transport system gets plugged up and we're not recycling NADH and we're not recycling FADH2, the first thing that's going to happen is the citric acid cycle is going to stop in its tracks because it needs NAD and FAD. Okay. Well, like I said, I show you this figure because it makes the problem look a lot worse than it is. I'm going to step you through it, and then I'm going to show you an easy way to do it. All right? So, NADH comes into the electron transport system and dumps its electrons off at NADH NADHQ oxidoreductase. And by the way, I wouldn't recommend writing this down while I'm saying it. You'll see why in a minute. Okay? NADHQ oxidoreductase dumps its electrons off to an enzyme or to a molecule called coenzyme Q, labeled here as Q. Coenzyme Q can also accept electrons from succinate Q reductase, and you can see it is a sort of a gathering point for electrons from either FADH2 or ultimately from NADH. Coenzyme Q dumps its electrons off onto Q cytochrome C oxidoreductase, which in turn dumps its electrons off onto cytochrome C, which in turn dumps its electrons off onto cytochrome C oxidase, which dumps its electrons off onto oxygen. Now, if there's one thing out of this cycle at the moment you want to write down, it is that electrons coming from NADH and or succinate Q reductase, which we'll talk about, which is where the FADH2 comes in, they dump off their electrons, ultimately ending up on oxygen and making water. So electrons in the electron transport system end up on oxygen, which is the terminal electron acceptor. When they gather those electrons and that oxygen gathers protons, we make water. So water is the final product. Okay? You feeling a little bloated? Maybe you're just doing too much electron transport. You know, have you thought about that? Maybe not. Now, this is too complicated. This is dumb. All right? This is dumb. And in fact, most books don't even bother with doing this because everybody has different names for these, and the different names make this process very, very easy to remember. And we're going to use the easy to remember things because you've got enough things to remember that are hard to remember. NADH dumps its electrons off onto this guy right here, and this guy right here we call complex 1. Simple. It starts at complex 1, at least for NADH's electrons. Yes, you should know complex 1. FADH2 actually is over here. It dumps its electrons off onto this thing right here, the succinate Q reductase. And guess what? We call that one complex 2. Okay? This guy is the same as the enzyme, the succinate dehydrogenase we talked about in the membrane of the mitochondrion in the citric acid cycle. Same thing. Complex 2. All right. Q is already short enough. It's kind of hard for us to make that any shorter or simpler. So we'll just call that Q or co-Q. All right? I'll let you guys tell me what we should call this. Complex 3. Okay? And that's what we'll call it. This is complex 3. Cytochrome C doesn't have another name. It's still called cytochrome C. So we've got a Q and we've got a cytochrome C. All right? And down here, of course, we've got complex 4. So that's the flow of electrons. Now, we see entry points for both electrons from both FADH2 and from NADH. And by virtue of the fact they come in at different places in the cycle, we will see that there's different amounts of energy that's generated as a result of the movements of electrons through these. The movement of electrons through these complexes is generating potential energy. Underline that. Okay? The movement of electrons through the complexes is generating potential energy. We'll see what the form of that is in just a minute. Okay, now, you see volts on the side. We're not going to worry ourselves about oxidation and reduction potentials. If you're curious, I will just simply tell you that we measure the potential, that is the voltage, as it were, of an individual complex. And complexes that have a more positive value will take electrons away from those that have a more negative value. So literally, this guy is sucking electrons down this way. Okay? Literally sucking electrons down towards oxygen. Okay. This, I think, is a better representation of what's actually happening. So what you see on the screen is the representation of the mitochondrion. Inside the mitochondrion is down. Outside the mitochondrion is up. And where it says intermembrane space, remember we've got an inner mitochondrial membrane shown here, and we've got an outer mitochondrial membrane. So the intermembrane space is that space between the outer membrane and the inner membrane. So this is the matrix. This is outside the mitochondrion, or outside the inner mitochondrial membrane. All right, so here's what you just saw. Citric acid cycle generates NADH. It generates FADH2. This makes it look like electrons go from complex 1 to complex 2, and that's not right, as you saw in the previous figure. This should have made a sort of loop around there to coenzyme Q. They both go into coenzyme Q. Then electrons go to complex 3. They skip cytochrome C, for reasons I don't quite understand, and then ultimately go to complex 4 and then to oxygen to make water. Now, this figure also shows how the potential energy is actually generated, because you can see as you look of the movement of electrons through complex 1, complex 3, and complex 4, that protons are being removed from the matrix out into the intermembrane space. Remember, we're making a concentration gradient, and we're making a charge gradient. It's going to be more positive out here than it is in here. We're going to have a higher concentration of protons out here than there is in here. That's potential energy that can be used to do something. And what we'll see in oxidative phosphorylation is that that potential energy is used to make ATP. It does not happen in the electron transport system. All that the electron transport system does is it makes the potential energy. You can think of the electron transport system as charging the battery. We're charging the battery. And then we make the battery do something in oxidative phosphorylation. Okay, questions about that? Okay, I'll tell you a little piece of trivia. One piece of trivia is right here. Coenzyme Q. We'll see how this works in just a minute. But coenzyme Q plays a critical role in this process. Electrons coming from NADH or from FADH2 come in pairs. 2 electrons. So when NADH dumps off its electrons, it dumps off 2 electrons. When FADH2 dumps off its electrons, it dumps off 2 electrons. However, complex 3, cytochrome C, and complex 4 can only handle 1 electron at a time. 1 electron at a time. That means that there has to be something, and I like to call it a traffic cop. This guy's a traffic cop. It accepts 2 electrons, and it passes them off 1 at a time. Another common question I get out of this figure is, well, where do those protons come from? Okay? Did something have to give up those protons for those protons to be there? And the answer is no. Remember, we're talking about an aqueous solution. We're talking about approximately pH 7. And at pH 7, you've got 10 to the minus 7th molar protons already there in any solution of water. So there's protons there that can be moved. We're going to move the protons out. That's going to cause the pH to drop up here and cause it to increase down here. So we'll see a concentration gradient of protons, and we'll see a charge gradient across that membrane, both of which will help us to make additional ATP. Okay. This figure simply shows us, and there's nothing on here that you really need to memorize for an exam or anything, but I show it to you to illustrate the complexity of these complexes. There's complex 1 right there. 46 proteins in complex 1. Okay? This is, when they say complex, they're not exaggerating. Okay? It really is a complex. It's the most complex of all the complexes, but even the simplest, succinate, the complex 2 has 4 proteins in it. Complex 3 has 11. Complex 4 has 13. Over here we see the groups that are involved in transferring those electrons or carrying those electrons. Okay? So we see flavins. When you hear about flavins being important, FAD stands for flavin, adenine dinucleotide. Flavins carry electrons. So FMN is a flavin mononucleotide. We see FES, Fe, of course, being iron. These early proteins here are called iron-sulfur proteins because they have iron bound to sulfur, and they participate in that electron transfer. The remaining guys are known as hemes and or cytochromes. Okay? So cytochromes are proteins that contain a heme group, basically. Cytochromes are proteins that contain a heme, and you've seen heme before in hemoglobin. It's a big ring complex. It's got an iron in it. And that iron now, instead of carrying oxygen as we saw in the hemoglobin, is carrying electrons in the electron transport system. All of the cytochromes, and the cytochromes start right here, all of the cytochromes contain iron except for these guys down here in complex 4. Instead of having iron, they have copper. And I'll say more about that. Okay. Now, there are things that are known as electron transport inhibitors. Okay? They're useful in a couple of respects. Some of these function very nicely as natural insecticides. Okay? Rotenone is a compound made by plants to kill insects. So you can buy at an organic garden shop, you can buy some rotenone. It's actually an insecticide that's organic. It's made by a plant. You spray it on your tomato plants to keep the bugs from eating them, and you're set. The way it works is it blocks movement of electrons through complex 1. So rotenone blocks the movement of electrons through complex 1. And by the way, you notice I'm talking about electrons and I'm talking about protons. They're doing very different things. Don't confuse them. Electrons move through the complexes. Protons get pumped by the complexes. So the electrons are the energy source for the proton pumping. Electrons are the energy source for the proton pumping. Okay, so you see another compound up here called amytal. Amytal is also an electron transport inhibitor. It also inhibits the movement of electrons through complex 1. And you might say, well, rotenone, that must be a poison. It must kill us, right? Why do we put that on our tomato plants? And while I wouldn't recommend going out and putting it on your cereal, probably not a good idea, okay, it's not as poisonous as some of these other things. And the reason is because, let's imagine right here I block complex 1. I can still get electrons in via complex 2. I've got an end around, right? In insects, this is a little bit different setup, and there's a different problem. We'll actually talk about insects later when we talk about insect muscle, okay? But that's more of a problem for insects than it is for you or I. If we look at complex 3, movement of electrons through complex 3 is blocked by antimycin A. This guy's more poisonous. This isn't something you probably want to mess with. There are ways around it, none of which we're going to talk about here. There are ways around it, but they're not very easy. This guy's a more severe poison than these guys are. Now, the most poisonous things on here block movement of electrons through complex 4. No surprise, there's our buddy cyanide. Okay? This is known as azide, N3-. And here's our friend carbon monoxide again. We've seen carbon monoxide get you two ways. It gets you in your hemoglobin because it can displace and replace oxygen, and it can stop electron transport. Carbon monoxide is not something you want to mess with. Now, if you block right here, everything plugs up. You might say, well, they flow here, 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 here. Okay? Well, yeah, they do, but they don't make it past here. Then this guy starts plugging up, then this guy starts plugging up, and this guy starts plugging up. And ultimately, when we do this, the whole system stops. And what happens when the whole system stops? NADH cannot transfer its electrons. FADH2 cannot transfer its electrons. And the citric acid cycle is going to stop. Okay? So now you see a connectedness between the movement of electrons through electron transport and the citric acid cycle. Yes, sir?
Does carbon monoxide use the same mechanism to block it up as it does with electrons, like attaching to it?
By attaching to the heme group? Yeah, that's a good question. I believe it does, but I don't know the exact mechanism in complex 4. I can't tell you that for sure. But it wouldn't be surprising that it would do that because you know it has affinity for the iron in the heme group. Okay. So those are inhibitors. Inhibitors are very useful tools in the laboratory for measuring how much energy is generated. We can actually isolate mitochondria in the laboratory and then block them with some of these things and study how much energy is generated when they're blocked. So that's a useful structure. Okay, I'm going to show you a couple structures, neither of which you're responsible for. Just show them to you so you know what's there. These are quinones, and quinones are the molecules for which coenzyme Q gets its name, the Q. The Q comes from the quinone. The quinone has a long hydrophobic tail, which allows it to stick in the membrane. This part sticks out of the membrane a little bit. And what you see is that this guy is capable of accepting one electron or two electrons and then passing them off one at a time. That's what this figure is trying to show you. That's the traffic cop function of coenzyme Q that I talked about before. Accepting two electrons and then passing them off one at a time. Okay. Flavins, you've seen these before. This is flavin mononucleotide, and the flavin part is this flat ring structure up here. And again, we see this is how we're making the FADH2. There's the two electrons and two protons. You're not responsible for structure, but you can see that we've changed from this structure over here to this structure over here. There's the two protons, and instead of having two double bonds, we now have one double bond, meaning that we've gained electrons. Iron-sulfur compounds. Iron-sulfur compounds are really surprisingly complicated. Okay? Surprisingly complicated. They can exist in a lot of different geometric forms. Notice how iron is being complexed by the sulfur side chain of cysteines. Okay? And we see this can be in a variety of geometric forms. Here's one iron, here's two irons, here's a complex of irons. And there's many other combinations that can exist for iron-sulfur proteins. Okay? So that's just an interesting sidelight about the chemistry of iron and iron-sulfur compounds. Okay. Now, what I want to do is sort of think up close and personal about what's happening in the individual complexes. So if we look at the individual complexes, what we're looking at here is complex 1. And no, I'm not going to hold you responsible for what's in here. But I want to sort of talk you through it just so you can realize what's going on. And remember that we're talking about a complex that has 46 proteins in it. And only the grand sum of 2 electrons are passing through it. Which means that these proteins, some of them carry electrons, some of them don't carry electrons, but they may support proteins that are carrying electrons. There may be passing of electron to complex, to complex, to complex, to complex, to complex before it finally gets out of the complex 1. I shouldn't say complex, but call this from protein to protein to protein to protein to protein before it gets out of complex 1. All right? And we're not going to go into those individual roles, so don't worry. But looking at it schematically, what we see is that we have NADH coming and dumping off its electrons. We're looking here in the matrix of the mitochondrion. Notice that NADH has to be in the matrix. I'll remind you later of that. It means if we're going to dump electrons off from NADH, we have to get NADH into the matrix. And though the citric acid cycle puts its NADH in the matrix automatically, glycolysis does not do that. Remember, we made NADH in glycolysis, and if we want to use those electrons, we've got to get that NADH in. That'll be a problem later we'll talk about. Anyway, NADH in the matrix dumps off its electrons. The electrons pass through several proteins, which are iron-sulfur proteins. Ultimately, those electrons are dumped onto coenzyme Q. It gains the electrons, and when it gains it, we write it with a QH2. Before it's gotten those electrons, it's a Q. So again, it's picking up 2 electrons at this point, and QH2 is in the membrane. Remember, because of the fluid mosaic model, these molecules in the membrane can move. And so coenzyme Q is moving around, bumping into other things in the membrane. When it bumps into a complex 3, it's going to donate its electrons. And we'll see how that happens next. Okay. This is the structure of complex 3. There are within these iron-sulfur centers, like we saw in the other one before. That's not of too much importance. We also see that complex 3 has the first heme groups in cytochromes that we've seen. Okay, so complex 3 has both iron-sulfur proteins and heme proteins. But more importantly, what happens in complex 3 is a cycle that we refer to as the Q cycle. The Q cycle, okay, works like this. So I want you to remember with that last, or a couple slides ago, where I had the QH2 that was being made. So QH2 is coming from the complex 1 or complex 2. The same thing happens in complex 2, where the Q is getting 2 electrons. This Q pool simply means that we have QH2. It's reduced coenzyme Q. It's got electrons, it's got protons. All right. Now, the Q cycle tells us how these electrons actually move through complex 3. And so I'm going to go through that in a little bit of detail. The process starts with the movement of electrons in. Okay, the electrons coming into the complex, and there's our QH2. That's our coenzyme Q with the electrons. And it does a divide and conquer approach. Okay? Coenzyme Q dumps 1 electron off to cytochrome C, which is docked on the complex 3. So it's docked. It's actually touching complex 3. There's cytochrome C. Remember, cytochrome C can only handle 1 electron at a time. There's the electron. The other electron that coenzyme Q was carrying is transferred to another Q that is being held inside of complex 3. So we've got a Q coming in, we've got a Q that's already there, and this Q that's already there did not have any electrons on it. So to account for our electrons, 2 electrons, 1 goes to cytochrome C, 1 goes to an oxidized coenzyme Q to get a Q with 1 electron on it. Everybody with me so far? Now, cytochrome C, of course, has gotten its electron, so it gets out of the picture. It's now going to take its electron and go pass it on to complex 4. So it leaves. Cytochrome C is going to leave. Meanwhile, the thing I haven't accounted for were these 2 protons. I've talked about the 2 electrons. What happens to the 2 protons? Okay? The 2 protons are dumped, and unfortunately, in this figure, up is into the matrix. Okay? Into the matrix is up. All right? What happens is this guy, the 2 protons that were here, are just dumped into the matrix. So we've already seen that we've had 2 protons that have moved from here up to here. Now, we've only done half of the cycle. Okay? The other half of the cycle, remember we still have this guy down here that's got 1 electron, right? We've got to get this guy ultimately back to no electrons, right? And so what we do is another coenzyme Q comes in. Another coenzyme Q comes in. The other coenzyme Q comes in, and it does exactly the same thing the first coenzyme Q did. It divides and conquers. So it transfers an electron to a new cytochrome C. We've had a new one come in the meantime. And it transfers the other electron to the Q that had 1 electron on it. Okay? And this guy grabs 2 protons out of here. We have a QH2. Guess what this guy's going to do? It's going to go back and join the pool. And a new oxidized Q is going to come in. This QH2, this QH2 becomes Q. It goes out here and, of course, can come back in as an oxidized Q over here. So what's happened overall? We've had 2 coenzyme Qs come in. We've made a total of 2 cytochrome Cs. We've made 1 coenzyme Q. So we've only done 1 net coenzyme Q. 2 came in, 1 goes out. Okay? And we've moved 4 protons into the matrix. The protons came from coenzyme Q. Now, this figure is always confusing, and I see on your faces, I see the confusion, so I'll stop for questions. Jenny? I mean, I think in the matrix. You're right, moving them out of the matrix, moving them out, sorry. Sorry. No wonder you have that look on your face. Moving out of the matrix. I'm sorry. Matrix is down here. Outside is up. So sorry. I said it completely backwards. So now that everybody's totally confused. All right. So the protons are coming from the matrix and going to the outside. Yeah. Does that make more sense? I'm glad I'm not taking a test today. You guys I would have failed it probably, and I would have gotten no mercy from anybody here either. Can I get partial credit, Jessica?
So I'm confused on the accounting. Is the second key coming from the FADH2?
So the Q pool. Remember, we've got millions of coenzyme Qs in the membrane. So that could come from FADH2, could come from NADH. There's no way of telling. Yeah. Yes, Megan.
Is the Q pool like a structure or
No.
just an accumulation?
It's an accumulation. So, as I said, in the membrane of the cell, of the mitochondrion, first place, we're going to have thousands of complexes, 1, 2, 3, 4. We're going to have thousands of coenzyme Qs, and we're going to have thousands of cytochrome Cs. So the pool simply refers to the collection of all of them. And some of them are going to have a QH2. They're going to have 2 electrons and 2 protons. Some of them are going to be Q, and so the Q pool is simply represented by all of those possibilities. Does that make sense? Okay. Yeah.
Yeah, I'm confused with the second half of the cycle. Like if the proton is coming from QH2 and then 2 protons coming from inside.
Yep.
So isn't that 3 protons here?
Say it again now.
So 1 proton is coming from the QH2 from the.
2 protons from QH2 are going right there.
Okay, it's going out, but
Yep.
it's not coming down to.
So these protons are coming from the matrix. Okay? Okay. Well, let me step you through the process real briefly, and then I'll move on. So, real briefly, okay, what we have, the main thing that happens is divide and conquer. 2 electrons, 2 protons come in. They split. 1 goes to cytochrome C, 1 goes to Q. Protons kicked outside the membrane. Start the process over. Divide and conquer. A new cytochrome C gets a new electron. The new protons get kicked out. And the Q that had gotten reduced now gets fully reduced because it gets a second electron and picks up 2 protons from the matrix. Okay? That's what's happening in the Q cycle. Okay? Okay. Complex 4 is a pretty important complex because it's the last complex through which electrons pass before going to oxygen. And as I said earlier, complex 4 is the only complex that has heme groups that contain copper. The only complex that contains heme groups that contain copper. And of course, heme is a part of a protein. We call that protein a cytochrome. Okay? Now, I'm going to show you briefly how electrons pass through there. It's not quite as complicated as the Q cycle. Okay? But I'm going to step you through it. Here's complex 4. All right? Here are, now before I tell you this whole process, I need to tell you what's going to happen. All right? So remember, we're starting with molecular oxygen. That's an O2. At the end of this, we're going to make 2 H2Os. That means we're going to have to get 4 protons, and it means we're going to have to get 4 electrons. All right? Each cytochrome C carries 1 electron. That means we're going to have to get 4 cytochrome Cs, and that's why you see 2 here and 2 here. Now, this process of bringing electrons in here, 1 at a time, probably causes some of the reactive oxygen species that are so hazardous to be formed. Okay? Anytime we do not completely reduce oxygen, we have a reactive oxygen. Oxygen is extraordinarily reactive if it's not fully reduced. Okay? So if we stop this process here, after this, we've got reactive peroxides. If we stop it where we've gotten too many, where we've got an extra electron somewhere, we might have a superoxide, something that's a real problem. Okay? We don't want to have that. But that's going to be a problem associated with oxidative metabolism. That's why, remember I said, when we look at mitochondria that are in old cells versus young cells, we see the mitochondria all beat up. Probably happens because of reactive oxygen species. Now, this is what happens. So let's think about this in some detail. So this is a schematic of the different heme groups that are present in the complex 4. 2 cytochrome Cs come in, and when those 2 cytochrome Cs come in, each one dumps off an electron, and this guy goes from being in the oxidized form to being in the reduced form. That's what the little red is to designate there. So these guys have each gained 1 electron. Iron has gone from +3 to +2. Copper has gone from +2 to +1. The next step in the process, molecular oxygen comes in. Now, there's not enough electrons there to completely reduce oxygen. So it forms what's called a peroxide bridge right here. This peroxide bridge, okay, one side bound to iron, one side bound to copper. Guess what? It's taken electrons from each one, and guess what? Those guys have just become oxidized. Then we bring in 2 more electrons. And as we bring in those 2 electrons, what happens is we break that peroxide bond. And notice that now we have an iron attached to an OH, and we have a copper attached to an OH. We've brought 4 electrons in. This guy is fully reduced. The only thing we need now to make molecular water are protons. The protons cleave those bonds, and water is released. Then the process starts all over again. Now, notice that we've got protons and protons that, again, are being taken out of the matrix. We are reducing the concentration of protons in the matrix, and as a consequence, we are changing the concentration of protons relative to the outside, thereby increasing that gradient. There's also, there are also protons that are pumped in this process. They're not shown on here. But complex 4 is also moving protons outside as well, contributing to that gradient. All right. Let's see.
Water released into the matrix, or
The water is released, I believe, into the matrix. Into the matrix. Yep. Okay? Now, this shows it a little bit better. You see protons now being pumped. You see 4 protons that are being taken to make water. The difference is, if I have 4 I take out here, and I put them over there, I've changed the concentration of protons by 8. -4 here, +4 there. Right? 8 proton change just by that movement right there. 4 protons lost here means that we've got a total difference from what we started of 12 protons. So just by the movement of those 4 electrons, we've changed the proton concentration by 12 over this complex. Okay. Questions on that before I move on? So there's this lady that goes out and she buys this piano. Okay? And she goes to this garage sale. I shouldn't say she buys it. She looks at it, and this is dilapidated old piano. Okay? And it looks absolutely like it's beat up, you know, and the guy at the thing says, you know, $30, it's yours, lady, you know. And she says, yeah, I don't know. And she plays it, and it sounds atrocious, you know. She says, I don't know. And he says, I'll tell you what. He says, what you need to do, he said, you need to go find Fred Abernathy. He is the world's greatest piano tuner. Okay? And she says, you know, he says, no, look up on the yellow page. I'm not kidding you, you know. And he works cheap. So she says, what am I out? $30, right? So she gives the guy 30 bucks. She takes the piano home, and she sets it up, and she looks in the yellow pages, and sure enough, there's Fred Abernathy, the world's greatest piano tuner, and he's just down the street. So she calls him up and says, I got this piano. I need to have tuned. He says, be right there. He comes over, and he's got his tools, and he does a little this, he does a little bit of that. And she sits down, and she plays on this piano, and it's like a Steinway. She's absolutely astonished. She said, that's amazing. You know, and she pays him $100 or something for the tuning. And she's just happy as she can be. She's got this gorgeous piano now. It sounds incredible. And she can play it. And she can have a lot of fun with this thing. Okay? Has anybody heard this joke? Oh, good. Okay. I'm making it up as I go. So, all right. No, I'm not. All right. So she plays the piano for a while. And 6 months later, this piano's starting to get a little, you know, it's not sounding as good as it was. And she says, I call Fred. So she gets on the phone, she calls up Fred, and he says, hello? And she says, yes. She says, I need you to come tune my piano. And he says, oh, your name? And she gives him her name, and he says, that sounds familiar. She said, oh, yes. She says, you tuned this piano for me, she says, 6 months ago, and it was incredible. And he said, oh, he says, I'm sorry. He says, I can't do it. She says, why not? He says, because Abernathy only tunes once. Opportunity only. When you have to explain. Abernathy only. I should have stayed with biochemistry. I never should have tried this. Okay, I'll just stop telling jokes. Is that better? I'll just stop. All of it, my feelings are hurt. I'm very sensitive, okay? All right, let's move on to something not quite so funny. All right? But probably more important, okay? And the more important thing is an enzyme that is a fascinating enzyme. It's an enzyme called superoxide dismutase. Superoxide dismutase. Now, superoxide dismutase, we have several of them in our cells. So we have different forms of the enzyme and so forth. And superoxide dismutase has long been known to be important in protecting against damage caused by reactive oxygen species. Now, I said earlier you can form superoxide by electron transport, and that's really not true unless you have an accident or something where an electron gets dumped onto an oxygen where it shouldn't. Okay? But suffice it to say that what the enzyme does is it converts superoxide, which is this guy right here. It's an O2 with an extra electron. It converts superoxide into a safer form, in this case molecular oxygen. Now, this enzyme is of importance because without it we are much more prone to oxidative damage. And one of the human diseases that's known to involve a problem with this enzyme is Lou Gehrig's disease, amyotrophic lateral sclerosis. Okay, there's a genetic form of the disease and there's a non-genetic form of the disease. And at least in the case of the genetic form of the disease, it's known that this enzyme has mutated and that doesn't function properly. That occurs in about 10% of the cases. Dr. Joe Beckman in my department, also a member of the Linus Pauling Institute, does research on this enzyme. Okay? And they know an amazing number of things about the enzyme, though they still don't have a complete understanding of the disease, the non-genetic form of the disease. Anyway, the way this enzyme works is as follows. What you see on the screen, the circles are the enzyme. So M refers to superoxide dismutase. This enzyme, in its mechanism of action, grabs that electron that's extra up here and becomes reduced. That's why it's going from oxidized to reduced. Then, the reduced form of the enzyme participates in a second reaction with a second superoxide. And now, instead of taking the electron, it donates the electron to the superoxide. Protons come in, and we form hydrogen peroxide. Okay? This leaves us back in the oxidized form. Now, who can tell me from last term the enzyme mechanism that this enzyme is going through? We talked about different mechanisms of catalysis. Who remembers from last term? The enzyme is going through two different states, back and forth. What? No. Two different states, back and forth. That's the hint, back and forth. Ping pong. The ping pong mechanism? What's that?
RT states?
No, ping pong. I just told you the answer. It's ping pong. The ping pong mechanism. Anybody remember that? No. We did too. We talked about ping pong. We talked about... Oh, man. No wonder you don't get my jokes. No. The ping pong mechanism, where an enzyme is in one state and it's converted and then it dumps something off. I talked about it with respect to transaminases. Okay. All right, that's fine. I give. All right. But suffice it to say, this mechanism is ping pong. The enzyme exists in one state, it exists in a second state. And then it goes back to the first state. So that's why it's called a ping pong mechanism. In any event, okay. Maybe I'm just the crazy one here. The bottom line of this is the concentration of superoxide in the cell is reduced dramatically. Now, one of the places we find this enzyme is in the lysosomes and peroxisomes. H2O2 is hydrogen peroxide. It itself is fairly reactive as well. But it's not as reactive as this guy is. And this guy can either be used by our immune system to kill cells. One of the mechanisms that macrophages use is actually killing cells with hydrogen peroxide. The other thing that can happen to this is that this can be broken down by an enzyme that we have very abundantly in our cells called catalase. C-A-T-A-L-A-S-E. Catalase can break that down. So what we've done with this enzyme is we've converted something that's very, very reactive, very, very much of a problem, to something that's much less of a problem that our other enzymes of the cell can handle. Make sense? Okay. So that's what's up with superoxide. There are other enzymes involved in protecting against reactive oxygen species, but superoxide dismutase is one of the very important ones. When we look at the medical considerations for reactive oxygen species, we see a very large number of them that have been linked in one form or another to reactive oxygen species. This guy right here, atherogenesis, which is atherosclerosis, plugging of the arteries and so forth, is increasingly being looked at because we now know that inflammation of the arteries is oftentimes leading to things like this, and inflammation can happen as a result of reactive oxygen species. And as I've said before, one of the ways that increasing your concentration of reactive oxygen species is to start smoking. That will definitely do it. Okay? So a real problem right there. Other things associated with it, Parkinson's, okay? Muscular dystrophy. What else? Well, you see, you can read the rest of them. I won't go through them. But in any event, reactive oxygen species are a problem. It's one of the reasons why research on molecules known as antioxidants is of a lot of interest. It's one of the main focuses of the Linus Pauling Institute here at Oregon State. And antioxidant substances are molecules like vitamin E, like vitamin C, that actually disable reactive oxygen species by reacting with them. So that's of a lot of interest and a lot of health interest today. Okay. Let's see. Where am I? I'm going to just say a word about oxidative phosphorylation, then we're going to finish for the day. Now, oxidative phosphorylation goes hand in hand with electron transport, usually. Now, oxidative phosphorylation is where that potential energy of the proton pumping is converted into ATP. It occurs in the mitochondrial inner membrane, as we saw before. And this figure is very unfortunate. Okay, it's accurate, but it's not drawn very well. What we see here is the inner matrix goes here, and then it goes all the way around. Okay, so outside is here, inside is all the way around. It's kind of like it's inverted. All right. Now, electron transport is pumping protons into the intermembrane space. And in the intermembrane space, what's happening is there's a complex called complex 5. It has several names. Complex 5 is probably the one you'll remember. But don't confuse it with the electron transport system because it's not part of the electron transport system. It's not part of the electron transport system. However, it is a proton channel. It allows protons to flow through it, and protons really want to flow through it. And when they flow through it, what happens is they turn a little turbine. That little turbine is shown right there, and that turbine generates ATP. So we're converting potential energy from the proton gradient to chemical energy in the form of ATP. And that's what's happening in oxidative phosphorylation. Now, I'll talk in some detail about that next time. It's good time for a 3-day weekend. See you guys on Wednesday.